almost start, still 15 more seconds, they told me, because they have to start the recording. Okay, I think I can start. Welcome everybody. I hope you enjoy the social dinner. Oh no, this is another story. My name is Alessandro Laio, and I'm chairing the first morning, well, this morning session. The first speaker of this morning is Professor Giorgio Colombo, who is Professor of Organic Chemistry at the University of Pavia and the head of Biocomputing Group. Giorgio, we look very much forward to your talk. You have half an hour, which will be followed by 15 minutes of questions. I will warn you when your time is almost finished. Please, Giorgio, go ahead. I would kindly ask all the other participants to switch off the cameras when the questions session will start. You can either raise your hand and then make a question directly, or you can write your question on the chat, and I will pass your question to the speaker. So please, Giorgio.
Okay, thanks, Alessandro, and thanks to all the organizers for this chance of speaking here. This is one of the first times for me to give a talk on Zoom. So I hope everything goes smoothly. I hope you can see my screen. All right. So what I want to talk to you today about is what we try to do in the lab to try and use protein dynamics and the study of protein dynamics and simulations to help us design new molecules with potential functions in biology. So one of the things we try to do is to develop new computational chemistry methods to investigate how a certain protein will interact and will recognize with partners. In particular, in cells, you might have a protein, a biomolecule in general, which has to interact differently with different partners to carry out functions, regulations, to carry out the tasks that are needed for the cells to survive, to proliferate, and to live correctly. When these functions do not work properly, diseases come up. And what we want to do is to develop small molecules that help us fix these functions, these malfunctions, or that help us understand what mechanisms and what types of surfaces or what types of interfaces are mostly used by proteins to interact with other partners. So we want to develop small molecules that, by modulating the dynamics, also modulate functions. And also we want to design mimics of these differential interfaces so that we can target the formations of these particular complexes, of each of these particular complexes, with selective precision and with such that we can relate the disruption of a complex with a certain biological effect. So in order to do that, we generally try to use molecular dynamics, which is a technique that you have seen described at length in the talks of Giovanni Bussi, of Alessandro Magistrato, and Alessandro Laio. So I won't dwell too much onto that. But what we want to do is to try and extract from this messy motion of the protein that occurs on short time scales, the summation of motions on longer time scales that is related to functional and functional aspects of how the protein works. So in this case, once we have understood the way a protein works, we can identify potential sites on the protein that, when targeted by a small molecule, will send a conformational signal around throughout the molecule that is related to a certain functional or biological response. And we generally try to do that by using as a test case a particular protein, which is a molecular chaperone that I will tell you more about later. Also, these types of motions are responsible for how a protein interacts with a partner. So different types of protein conformations might select different partners by reshaping the potential interface, the protein might interact with different partners at different times in the cell cycle. So what we want to do in many of these cases is to try and predict which are the potentially interacting parts of the proteins and then mimic them by interaction mimics, which are synthetic molecules that we synthesize, in general in the form of peptides. So I will first start by trying to give you an idea of what we have done in trying to design these functional, small molecule functional regulators of protein functions. So as I said before, what we want to do is to devise or to design small molecules that by binding to sites alternative to the active site will change the dynamics of the molecule of the protein so that we obtain a modulated response or a different response than the response that you would obtain normally in the absence of the synthetic molecule. We applied these concepts to the study of this molecular chaperone, which is called HSP90, which is a very complex protein composed of two monomers. It's a dimer composed of these two monomers, whose functional dynamics, you see, is very complex and goes through a cycle of ATP binding, which brings the protein to the closed state, like a big molecular clamp that closes. And then this molecular clamp acts on other proteins and folds them to the correct and functional state. So once these other proteins, which are called clients, are folded, they will carry out their function. In cancer, these molecular chaperones, and in stress conditions, these molecular chaperones are highly overexpressed. And in general, they help cells, like cancer cells, which normally are living in a hostile environment, to adapt to that environment and to survive to that environment by overexpressing, by helping the overexpression of, say, kinases, glucocorticoid and hormone receptors and so on, which are needed by cells to survive. So if we can interfere with this type of mechanism, then we have a good chance to interfere with cancer pathways. HSP90 is very ubiquitous in cells. It's in the cytoplasm, and lately it has been found to be present also in the mitochondria, and the mitochondrial homologue is called TRAP1. And the interesting thing is that TRAP1 has got this interesting asymmetric dimeric structure, which crystallizes in this asymmetric dimeric form, which is different from the symmetric structure that you see in the cytosolic homologue. What is also interesting is that in some kinds of tumors, this TRAP1 is highly overexpressed. In other types of tumors, TRAP1 is highly downregulated. So what you want to do is to develop molecules that might target TRAP1 only where it is overexpressed without interfering with HSP90, which is normally used by other cells to carry out, to survive. So in order to do that, one strategy would be to target the ATP binding site, which is shared by all these chaperones. But as you know, ATP binding sites are very conserved, so you wouldn't be able to target TRAP1 while leaving HSP90 unscathed or untouched. ATP sites are conserved, ATP is very abundant, and so it's very hard to compete with it. But this type of asymmetry here gives us a chance to think about possible targeting of these sites by small molecules that can differentiate between this structure here and this structure here. So the question now becomes, how do we find a druggable pocket in here? Which is the most likely druggable pocket in this region that might interfere with even with ATP binding and then the functions of the whole protein? So let's go back to the mechanism. So the mechanism is the following. So ATP binds, the protein gets into the closed state and into this sort of asymmetric state, okay? And then one ATP gets hydrolyzed, this region here changes its conformation and reshapes the client protein in a mechanical way. So what we want to do is to find the residues in this region which are most responsive to ATP binding and hydrolysis, so that if we put something in here, if we put in like a wedge or a small molecule in here, we can send a conformational signal back up, interfering with ATP signaling in TRAP1 exclusively, and then interfering with mitochondrial processes that are at the basis of tumorigenesis. So in order to find these residues, which residues we need to target in this region, then we need to find what is it that communicates best with ATP and the ATP site. So in order to do that, we use this simple coordination propensity measure, which we developed a few years ago together with Christian Micheletti also, in which basically what you look at is the low distance fluctuations between every residue I and every residue J in the protein. And then when we focus on which residues in the protein are mostly connected to the residues that are in the ATP binding site, what we can see is something interesting. So if we consider the protein in the 2 ATP state, you can see that the protein is kind of highly connected, it's very rigid. These are the residues connected to ATP site for most of the simulation. And you see that these sort of allosteric communication pathways go down to the far domain of the protein, down to this region in both protomers. When ATP is bound to one arm, to the buckled arm, and ADP is bound to the straight arm, then you only see this region here that is highly connected to the ATP binding sites. And when you switch ADP for ATP between the buccal and the straight protomers, then you also see this region here. So this region in the middle part of the protein always appears to be involved in some sort of strict or strong communication with ATP binding sites. Interestingly, this region here for TRAP1, if we look at this measure calculated locally, so only on short stretches of residues, and we compare TRAP1 versus the homologous residues in HSP90, the red versus the black lines, you can see that the local dynamics also of these residues is very different between TRAP1 and HSP90. So these residues may be representing a sort of point of differentiation between the two proteins. So we can plot them onto the three-dimensional structure and then use a collection of structures that we sample from the simulations, from MD simulations, to look at the possible structures and conformations that need to be targeted by small molecules and that we can actually use to host small molecules. So we look at these residues, the ones that are most connected and that are related to the presence of a possible pocket that is small enough to host a small molecule, a drug-like small molecule. And then what we do is we look at what kind of functional groups these residues display to possible potential binding with a small molecule. We do that by developing a sort of pharmacophore model in which we ask the model to display a certain functionality that is complementary to the one presented in the site that we want to target. So let's say the protein presents a glutamate structure, a glutamate functional, glutamate side chain with a carboxylate function. Then in our pharmacophore model, we'll need to have a charged, positively charged group to form a plus-minus charge with the glutamate, which will give us a good electrostatic interaction. If we have hydrophobic interactions here, we also present hydrophobic interactions and so on. So once we have this model, we can use this model to screen for small molecule databases, which are available, for instance, from the Zinc database, or you can screen for commercial databases. And we did that, and we came up with about 35 candidates, 11 of which you could buy and test, and of which we first looked at the possibility for them to block ATPase activity, to block the hydrolysis of ATP in TRAP1 versus HSP90. So what you can see here, in blue we have TRAP1 and in red we have HSP90. You can see that the blockage of the ATPase activity always happens with the small molecules in TRAP1, but not in HSP90. So we have sort of selectivity versus the isoform that we are interested in terms of targeting the mitochondrial species. In some cases, even some compounds are inhibitors for TRAP1 and activators for HSP90, but we still have to look into that. Also, with these molecules, we also have a nice dose-response profile with IC50s that are in the low micromolar, which is not so fantastic for a drug, but is very good for a chemical tool that we can now use to mimic the effects of shutting down selectively TRAP1 while leaving HSP90 functioning in the cells. So this is what we did in collaboration with Andrea Rasola at the University of Padova, and he did the following experiments. So we know that when TRAP1 is active, it will bind SDH, succinate dehydrogenase, which is a protein involved in respiratory phenomena in the cell, and by targeting, by being activated, it will act as a brake on SDH. So if you ablate TRAP1, you knock down TRAP1 or you inhibit TRAP1, succinate dehydrogenase is released from the complex and becomes activated, and its activity will go up. So TRAP1 ablation will bring up SDH activity. And they did that by using a CRISPR-Cas technique. So when we look at the effects of the molecules, you can see that by using a normal inhibitor, which doesn't distinguish between TRAP and HSP90, you have, of course, this is an ATP competitive inhibitor, you have the activation of SDH the same way as by using TRAP1 ablation. This orange curve is TRAP1 ablation by biology. Here on the different lanes, you have the effects of the small molecules, all of which sort of produce a phenocopy, a copy of the phenotype introduced by the ablation and the knockdown of TRAP1. Also, when we test these molecules on tumorigenesis, you can see that while tumors grow nicely in the presence of TRAP1, in the presence of the small molecules at higher and higher doses, they will basically, this foci of tumor growth will disappear. And the SDH effect is also then observed in vivo in zebrafish larvae models. So we have a nice chemical tool, molecule 5, to start doing more experiments to investigate the functions of the proteins, their differential functions of two very highly homologous proteins by doing more experiments and by developing this molecule 5 even more using the models that we have generated so far. So to summarize this part, we have exploited structural and dynamic asymmetry as the basis to discover new isoform-specific allosteric inhibitors of TRAP1. It is the analysis of the nucleotide-dependent motions that allows us to identify the structures that are probably mostly involved in regulating these motions, and then that we can target to perturb these motions. And we, from the therapeutic point of view, we have selectivity, versus one isoform, which is highly overexpressed in certain types of tumors and not expressed in others, versus the various isoforms of HSP90, which is present all over the place, and you don't want to inhibit it indiscriminately, not to have side effects or off-target effects. And then we can use this data that we have generated for a modulation, for the mechanism rationalization and so on, to define novel leads and to design new ligands. So this brings me to the second part of my talk, in which we would like to interfere with the activities of these chaperone proteins by blocking their interactions with other proteins. So far I've talked to you about the way that the chaperone interacts with ATP and goes through the cycle as an isolated molecule. But in reality, it will go through this cycle by interacting with a number of other proteins, forming multi-protein assemblies, which are highly dynamic and which involve different players. So HSP90 binds ATP, gets into the sort of closed form, and then a co-chaperone, which is called CDC37, binds up and brings with it a client protein indicated by Cl here. The client is typically a kinase, and by bringing the kinase onto the complex, it will start the way that HSP90 works on the complex to reshape the client. So, these are very dynamic complexes, dynamic structures, and they have been finally shown in their full atomic detail by cryo-EM, by the group of David Agard a few years ago in a great science paper, which they showed that the HSP90 here, this X in the middle, binds to CDC37 that goes throughout all around the protein, all around HSP90, and to a client protein, which is here, which is a kinase. What's interesting here is that the kinase is sort of unfolded, and a part of it goes through the lumen of HSP90, and these large, long two helices are part of CDC37. So we have this client which is unfolded, partially unfolded. We have the co-chaperone, we have the full-length chaperone, HSP90. So how do we interfere? And this is a general mechanism that would involve other clients, other proteins, because HSP90 really acts on many other proteins. So how do we develop ligands or reagents that selectively detach one client from the protein, from the chaperone, while leaving others untouched? So and once we can predict how to do that, what do we design? We should design, unfolding region mimics. So this is the region that unfolds here. We need to find which regions are unfolding or are most prone to unfolding, and then we should design mimics of these regions, so structural mimics or sequence mimics of these regions, which can adapt to the surfaces and interfere with the process of full assembly. So what we did then was, okay, we don't know the unfolding pathway of or the full unfolded state or of complex proteins like kinases or hormone receptors and so on. So what we can do is to find the regions on these proteins by computation that are most prone to unfold. So in other words, the regions on the protein that are not so well connected or energetically coupled to the most stable regions of the client. So imagine this is a kinase or a receptor which has to bind to HSP90. One part of it will fold automatically by itself and will constitute the folding core of the protein. One part, which is the one that may be in contact and may be recruited by chaperones and co-chaperones, is not so stable and will be, of course, recruited by the co-chaperone and blocked by HSP90. So we need to predict this. How do we do this? We do this by predicting which regions on the client protein are most likely to be uncoupled to the folding core of the client. And we do that by running MD simulations of the client in isolation. We calculate all the possible non-bonded interactions between all the residues. Then we simplify this matrix by eigenvalue decomposition, and we filter it by the contact matrix. And we isolate all the couples, all the pairs of residues, which are on the surface or close to the surface of the protein, and that are not well coupled or not strongly coupled to the interior of the protein. For us, these regions, which we call epitopes, are the regions that are most likely to undergo unfolding because they are not strongly attached to the rest of the protein. So when we applied to kinases, for instance, we decided to apply it first to this ABL kinase, which is a tumor driver, and is different from the kinase that we have seen in the agar structure. We predict this region here in the end lobe in red as the region that is most likely or most prone to undergo unfolding transitions or to undergo unfolding. We don't know anything about the unfolded state. We just predict that this part of sequence in red will be the most likely to undergo unfolding transitions. So when we look at the complex of HSP90 with the other kinase and we superimpose the folded parts, the C-terminal domains of the original kinase in the cryo-EM structure 2 hours, you can see that the predicted epitope really traces the surface of HSP90 that is really then in touch with the unfolded part. So if we can realize this red part here in the form of an isolated peptide synthetically in the lab, and we then throw the peptide into the mixture of this chaperone plus the rest of the chaperone machinery, we should be able to break up the chaperone machinery and block its action on the folding of ABL. Okay, so that's what we do. We realize peptides that mimic these regions by synthetic, normal synthetic methodologies, and then we test them with NMR. We put in fluorinated fluorinated residues to run fluorine-19 NMR spectrum. And so this is the peptide that we realized mimicking this region in isolation. When we add HSP90, there is a shift of the signal indicating interaction. And then when we use this peptide to detach HSP90 from ABL from HSP90, you can see that ABL is not present in the complex anymore. Here you have HSP90 in the presence of the different sequences and different variants. Other proteins, CDK4, which is the original kinase, is not touched, is always there. But ABL, in the presence of the different peptides, is completely absent in the cells, is not folded anymore, as well as this co-chaperone AHA1, which is detached from the complex. Then, this is one case to check whether this could be generalized to others. We carried out our predictions on other kinases, like BRAF, which is hard to drug. It's one of those undruggable proteins, CDK4, SARC, GR1, etc., which are all related to tumorigenesis. We predicted the regions, and then we decided to also synthesize them as synthetic peptides and look at their interactions, again, with fluorine NMR. The different lanes indicate the different peptides. These are the signals to binding to CDC37, AHA1, which are co-chaperones, and HSP90. So you see the different peptides may bind to different parts of the chaperone machinery, to different players in the chaperone machinery. And then what do? They do exactly what we want. So they detach their cognate protein, BRAF, in the case of the BRAF peptides, CDK4, SARC, and so on. What's interesting also is that some of the peptides, in particular,
CDK4, 3-4 more minutes.
Yeah,
3-4 more minutes. Right.
I'm done, basically. So this CDK4, for instance, induces the expression of this caspase, which is related to apoptosis in the cells. So by disrupting these complexes, we are also inducing apoptosis and cell death in HSP90 dependent mechanism. So we basically have found a way to target selective and difficult protein-protein interactions to induce the degradation of difficult to target by small molecules proteins. And we also have a number of different sequences that we can use to evolve into potential better chemical tools or even drugs, but that's a much longer way to go. To just summarize, so it is possible then to combine computational chemistry methods, the analysis of biological mechanisms in biological chemistry, and develop new chemical tools to combine all these three areas and develop molecules or small molecules that help us play around with biological systems and reshape their functions or better dissect the intricacies of the functions of these systems. So I got to the end, hopefully in time, and I would like just to thank all the people that work on this project that you see in this slide, all the collaborators. In particular, I'm really grateful to Andrea Rasola and Carlos Sanchez Martin at the University of Padua, besides all my group. And I would like to thank all the finance, all the funding, and all of you for your attention. So I'll be happy to take questions.
Thanks very much, Giorgio. Thanks very much also for staying really strictly on time. So start with questions which started appearing in the chat. I will directly make the questions just to save a little bit of time. So the first question is by Davide Bassani. The question is, what is the length required, the required length of a DMD simulation which is necessary to depict the allosteric activity of TREP1?
Okay. So these simulations have all been done in multiple replicas and are in general between half a microsecond to 1 point something microseconds each. So for each system, we have carried out pretty extensive simulations in replicates. And so we try to filter out only the signals that are consistently consistently coming up. In the original paper of the TRAP1 study, we did also simulations were still a little shorter than that, which we used then for the compound screening. And however, we used different force fields also. So we used Gromos and Amber force fields to compare the data and the results, and they were, overall consistent.
Okay, then there is a second question from some
for the screening of compounds.
Yeah.
So the question is, for the screening of compounds, did you use flexible docking program algorithms? So I'm sorry, I haven't been very clear on that. We don't use docking. Okay, so we try not to use that, at least at the beginning for the selection of the compounds. So once we have identified the site, I don't know if I'm trying to use my hands, but once we have identified the site where the molecule could bind, we look at the chemistry and at the conformations of the potential binding site. Then what we do is we make a sort of mold of this binding site, and we look, when we look at the chemistry, we identify the hotspots that are needed to bind. So if you have a glutamate, you will need, you will have on the protein, it's a negative charge on the protein. So you'll need on a molecule a positive charge to bind there. So we just represent that as a function, as a plus charge in one position with a certain distance from, say, another point which has another characteristic complementary to the potential binding site. And then we screen these for, we use this pharmacophore model to screen for molecules. And then we only do docking afterwards, at the really end of the process. Now we are studying the small molecules within the protein context, and we have used flexible docking algorithm to put the small molecules in to have good starting structures.
Then there is another question which is more technical. Which tool do you use for pharmacophore predictions? So
in this case, we have used a combination. We are very deterministic on that, so opportunistic also. We used both the ones available in the BioVia programs and the ones available in Maestro. And we try to generate a consensus of the different algorithms, and generally they work pretty well. One thing that is also important is some sort of chemical sensitivity. So, you really want to avoid having large, huge hydrophobic areas, which will make your molecule insoluble, or too many different stereogenic centers and so on.
Clear. Yeah. Okay, so the next question is more general, and it's also connected to another question that I had myself, is, well, can you please explain again how to compute the coordination propensity and how to exploit it for comparison of two proteins? My question, which was related and I wanted to ask, is also if you can generalize this measure to take into account somehow the causality, in a way, if a specific contact is responsible for the change of another contact somewhere else in the future.
In this case, not really. These are on average properties from the trajectory, so we kind of lose the causality.
Yeah.
We are trying to look at the coordination propensities on different length scales of the simulations and trying to analyze them using different types of techniques, but it's kind of hard.
But going back to the question, which is more general, maybe you can just...
Okay, so what we do is we calculate from the simulation, you have every possible fluctuation between the atom I and atom J, so they have a distance. So these distances fluctuate, right? So we decide that two residues are in communication, mechanical communication, if their distance fluctuation is below a threshold, which is defined on the basis of a local parameter. And then we find all the pairs of residues that have this fluctuation in the distance below this threshold, which is typically the average fluctuation that you calculate between every residue I and I+4, which is one turn of a helix, basically. So if you have high coordination at for residues that are very, physically separated, that might not be a trivial signal. That might be something related to how the protein is packed and how different secondary structures are organized to transmit a signal from, say, an active site to another part of the protein. How do we use it to compare two proteins? You can calculate it for different proteins and then realign them by the sequence. And then we can either calculate the sum of the columns of the matrix that we obtain for the coordination propensity matrix, or look at transform it into a local fluctuation parameter like the one I showed in talk, and then look at the realign these graphs on the basis of the sequence alignment so that we can compare two or more different proteins. Yeah, that's what we do.
Yeah, so actually there are three more questions, all related to the coordination propensity and much related within each other. So I'm going to read these three questions at the same time, and we can see if in the last 3-4 minutes you can give us a hint. Well, could the coordination propensity be useful for evaluating, well, for discovering potential regions of the protein for allosteric inhibition? Question
That's
number
exactly
one.
the point.
Well, the second one, is there any connection between the coordination propensity and conformational thermodynamics? Question number two. And third question, still also on the same topic, discovery. Well,
yeah, the threshold I just
the threshold you just mentioned. So this you have
mentioned. The coordination propensity and conformational thermodynamics. It's probably something qualitative that we can say at the moment. So if if you have regions that are highly coordinated or that constitute highly coordinated domains, you may imagine that these move in a how do you say, in a coordinated way as well, or in a I missed the word. I'm sorry.
Concerted.
In a concerted, exactly, in a concerted fashion. So you may extend it to identify some sort of, to predict some sort of overall or large scale motion of all these regions that are conserved, or you can display, I mean, if they are subdivided by flexible regions or unconnected regions, you can imagine that these unconnected regions will be hinges, but we cannot really use it to really get any quantitative data on conformational thermodynamics. It's more qualitatively, really.
All right, we still have really 2 minutes, 2 minutes. There is a question which is not related to that. Is well, since most of the pockets that you find in your analysis are highly flexible, how do you actually choose the shape of the pocket on which you perform docking? How do you?
We actually really do not perform docking to screen for the compounds, but we use different pocket conformations that we sample from the simulations to develop adaptive, let's say, pharmacophore models that are complementary to the different shapes of the pocket that we identify. And then we try to build a sort of a consensus pharmacophore that recapitulates all the chemical functions that are consistently required in all the different structures to bind productively to all these different structures. I don't know if that's clear.
I think it's clear enough. Well, actually, Giorgio, now I think we have to move to the next speaker, but there are other, I think 2 or 3 questions which I didn't mention, which are more general, like MD details. So you can maybe reply directly on the chat.
So there's references in the talk, so you can find the methods and the coordination propensity.
Also for they want a publication about the coordination propensity. So
you just email me and I'll send you the links and update.
So I clap my hands on the behalf of everybody, and I think we can now move to the next speaker, who is Marco De Vivo. Marco De Vivo is working at IIT, Italian Institute of Technology. He's a research lab director and working on well, molecular modeling and drug discovery. So topics which are very much related to the issues which were already discussed by Giorgio. So Marco, please go ahead with your talk.
Hi, good morning, everybody. Thanks, Alessandro, for the introduction, and thanks to all the organizers for the invitation. I really do hope to meet you all in person soon, and maybe for a second edition of this nice workshop and school. Okay, so in the spirit of the title of the conference and talks I've seen, you will see in my talk a number of examples where I think we've put a good use in molecular dynamics, either classical or quantum, to study biological processes that are quite challenging. So actually, oops, Okay. In the group, we have a number of interests on pharmaceutical relevant targets, going from membrane-bound proteins actually up to functionalized gold nanoparticles. And what I wanted to mention is that everything we do here is in terms of computations and molecular dynamics mostly, but then we translate this knowledge into the actual synthesis of small molecules within the group. So we are lucky in a way, so we can put our information into the rational design of small molecules that are used then to initiate drug discovery up to in vitro in the group when possible. And then of course we rely on external collaborators to move to animal models. And before starting the talk, which today will be on, as you know, DNA and RNA processing metalloenzymes, I'd like to have a snapshot on the drug discovery part of the group, which has been quite active over the last few years with a couple of lead compounds that are active in vivo in neuroscience and cancer, as you see, and we hope to move forward these compounds in advanced preclinical models very, very soon. So just to give a snapshot of how then we use molecular dynamics. But today we will stay on computations on molecular dynamics of these type of enzymes that process DNA and RNA. Actually, we look at a bunch of them in terms also classes. So these are polymerases, nucleases. So polymerases, elongation of nucleic acids, nucleases, they cut nucleic acids, ribozymes, which are RNA enzymes, RNA-based enzymes, and topoisomerases, which regulate the topology of nucleic acid DNA. Now, all these enzymes, amazingly, have conserved the same catalytic site, the same architecture of the catalytic site, which is a so-called two-metal ion active site. So all the drugs that act on the catalytic site of these enzymes target the same type of architecture. Now, this is important because it goes back to a seminal paper that was published years ago by Thomas Stites, a Nobel laureate, and his wife, Joanne Stites. It's a very seminal paper on this particular mechanism that I suggest those young in the audience to read, because it captured all the essential aspect of this mechanism based on the very first structure where they saw this particular architecture, where there is one ion that stabilizes the nucleophilic oxygen, the other ion that stabilizes the leaving group, and they together stabilize the transition state for either for the forceful transfer reaction. That's the name of it. Okay, so I started to look at this type of action times ago, and the approach basically that we use is the following. We first study through classical MD the targeted enzyme, and for this, of course, the challenging aspect is the metal-centered catalytic site, where we use a non-bonded approach for the metal ligand interaction. We have a redistribution of the charges skin to redistribute the charges to stabilize the site at times. And then, of course, once we want to look at bond forming and breaking events, we have to switch to quantum simulations. In most of the cases, we use the Carpernello coordinates QMMM implementation, as Alessandro, in fact, implemented with Ursula years ago. And we still use that in most of the cases. And of course, then we have to couple these simulations to enhanced sampling techniques, where the trick is always to find the right coordinate, reaction coordinate or collective variable to describe your reaction, the reaction you are interested in. Now, how do you know if the profile you get out of this enhanced sample simulation is the reasonable or a possible pathway of your catalytic mechanism? Well, there are three criteria that you have to satisfy. The first of all, of course, you want to see the reaction that you have in mind. So you have to generate the product as expected, as you know it has to be formed. Second one, the free energy barrier for this reaction to happen has to be better, meaning lower than the uncatalyzed reaction in water, of course, because you want to see the catalytic effect of the enzyme. And third one, you can compare the energy you got out of your enhanced sample simulations with the K-cut. And then if all these criteria are satisfied, you can say with a reasonable confidence that your reaction mechanism is a potential one and the correct one. So we have used this approach in a number of cases, starting several years ago on a ribonuclease H catalysis, because it is a 2-metal ion site and it is a promising target for HIV. We have compared this mechanism with a 1-ion mechanism, another enzyme, same reaction, but with only 1 magnesium. We have had a paper years ago where we proposed the presence of a 3rd ion, and you will see this is quite relevant for the following of the presentation. This was predicted only based on MD simulation at the time. Then topoisomerases, and more recently, DNA polymerases. And this is a particular polymerase that we are very much interested in, and I'll tell you more about that. Okay, so but let me make a point that is going to be useful for the rest of the simulations, for the rest of the presentation, sorry. So we try to integrate our view on taking into account the structure, the dynamics, and the function for catalysis of RNA and DNA. So this is a snapshot I took a few days, yesterday or the day before, from the PDB data bank. And this is the amount of structural information we have in the PDB data bank. Now, we are used to run a few model systems, but actually you see we are over 170,000 structures deposited in the PDB. And in particular, in my case, because of what I'm interested in this case, protein nucleic complexes, there are about 8,000, more than 8,000 complexes, including those very amazing complexes that now are resolved with cryo-EM. So we have to take advantage of this information, apart from those few that we can actually simulate. And that's the point I want to make here, and showing you a few cases where we generated mechanistic hypotheses based on an analysis of this information available today. Okay, so the very first example regards the DNA and RNA polymerases. Now, the catalytic site, the catalytic cycle that is here actually reviewed in this recent perspective, if you're interested, starts with the deprotonation of the 3' OH, and then there is the incoming nucleotide that has to be attached to the growing strand of the growing filament of DNA here, and the nucleotide has to be added. So I'm gonna go fast on this because then I want to reach the more recent stuff, but this is very interesting. So we looked at all the ternary complexes in the PDB data bank, which means the complexes that have the enzyme, the protein, of course, so the enzyme, the DNA, and the incoming nucleotide. And if you look at all the possible ternary complexes, so from prokaryotes, eukaryotes, viruses, you will always find that the entering nucleotide, which is this one here, has this particular conformation, which is not the conformation that has been reported in solution. It likes to stay linear with 1 magnesium ion. It doesn't fold in this way, forming this intramolecular hydrogen bond. Now, this is also this is a collaboration at that time with Paolo Carloni in Germany. And if you overlap all these incoming nucleotides taken from the ternary structures available in the PDB, you will see this particular conformation where you have always the formation of this intramolecular hydrogen bond. So this triggered at that time, this unprecedented observation, which was not reported, it triggered our proposal for a new reaction mechanism in order to have the addition of this incoming nucleotide to the growing DNA. So the reaction is this one. Here we have to form a new bond and break this. So I will jump to a movie where we have cut and paste. So it's a merge of classical and quantum simulations. This is the starting system equilibrated in MD. So you see here, now this is the nucleophilic oxygen, the protonated, this is the incoming nucleotide, and this is the intramolecular hydrogen bond that is formed and is stably maintained there, and which helps this conformation to be kept within the catalytic site. At this point, the quantum simulations, this is QMMM, so this is the QM part that you see in bold in licorice. There is the reaction happening, so this is the classical SN2 reaction with the inversion of the umbrella for the phosphorus transfer reaction. Here the proton starts to be shared, kind of shared, but of course there is no proton transfer because of pKa reasons, so the proton remains on the sugar, but at this point this proton will become this one here, so this has to partial translocate, and when we have this partial translocation and we switch to QM, then thanks to the presence of the magnesium, which lowers the pKa oxygen, then we can have the deprotonation. And here you fall within 2.5, I think, angstrom from the co-crystal of the product state. And so here we see that the protonation and partial translocation is a process that is concerted, and this is how we explain now this mechanism, which is a closed loop of sequence of chemical steps, so nucleophilic attack, proton transfer, and physical step, the partial translocation that leads to the addition of the incoming nucleotide. And we call this the self-activated mechanism because of this intramolecular hydrogen bond. So this is an example of how we used this information available in the many polymer structures that we had to generate a mechanistic hypothesis. Another example is a collaboration with the structural biology group of Marco Marcia, the MBL, with whom we put together a curated data set of structure with the integer metal ion at the center. And this includes over 130 enzyme, 49 3D structure, including the ribozyme, so the RNA based enzyme, nucleases, polymerases, and then we compare structurally what they conserved around the 2 metal ion that was still present in the structural alignment. And what you see here is that there are preserved spatial localization where positive charges, in this case is the ribosome, so there are no amino acids, and we have a potassium ion here and a potassium ion up here. These are the 2 magnesium ion, and where you have in the RNA-based structure, these 2 ions in proteins, you have lysines or arginines, okay? So there is the evolution replaced in a way these potassium ions, this positive charge with lysines or arginines, which are preserved in this particular position. And these are nucleases, including Cas9 and CRISPR-Cas9, and polymerases here. Of course, then you have to, you should see the overlap, but another way to look at this and the conservation of these positive charge, which are second shell positive charge around the 2 metal ion, is here. So these are a sphere of 4 angstrom where you have K1 and what we call K1 and K2, and these are about 4 angstrom, 5 angstrom from the substrate. Okay, so they are in touch with the substrate either on one side or on the other. And so in this way we have an expansion of what was believed to be only the 2 metal ion site, and with additional positive charge that are located in the surrounding of the conserved in the surrounding of the metal ion center. And this is a plot where you see the distance from the substrate. So you have restriction endonucleases, other nucleases, polymerases, and the 2 ions are always within a distance for an H bond basically from the substrate, so 3 angstrom. Now we have taken our enzyme. At that time we were still interested, and we are still interested in fact, in polymerase eta, and we mutated these 2 particular residues. And what you see is that indeed in the reactant complex, if you go from the wild type to either K1 or K2 or both are mutated into alanine, then you have the disruption of the optimal Michaelis complex with the deviation for the optimal number of Watson and Crick H bonds here that is present in the crystal structure, but also what we call the D stands for distance, new bond, which is the distance of the new bond information. So you have an elongation of the over the right here of the reaction coordinate, which means that it's less prone to undergo catalysis. Of course, we cannot look at the barrier in this case, but at least we see the stability or not of the Michaelis complex. What's useful for this for also, for example, here is a complex structure of the spliceosome, which is related to the ribosome, and you will see here that in this particular structure, there is a paper where they report that where the response to potassium binding on the spliceosome remains to be elucidated here on the bottom. So they don't know where the potassium here, but the potassium is needed in order to have the catalytic action. And here, if you use our model, you can place a potassium ion there, which fits quite nicely, and of course maybe was not there because of the resolution of this particular structure. So that's another example. Let me give you another additional example. Now, this is one of these particular residue that is conserved. This is actually a first shell, meaning that is in direct contact with the nucleotide, with the entering nucleotide, this residue here is either an arginine or a lysine. And these are the alpha carbon of this residue in a multitude of PDB that we have retrieved from our collection of structures of the ternary complex. And you see here all the enzymes, and these are 28 representatives in the figures here. And this particular residue was actually already known to be important for the chemical step. If, for example, in the case of polymerase eta, it is an arginine. It can be in different conformation, and depending on the conformation, it helps or not the catalytic step, stabilizing the transition state. And this is also what we have seen for the A conformation pointing down versus the other type of conformation. So we know the electrostatic effect of this arginine is important for the reaction to happen. What, however, we noticed looking again at the structural information available to us is that there are a few structures of polymerases that are reported in paper like in this one in Nature to have the Uxten base pairing instead of the Watson and Crick, where one of the bases flipped 180 degrees, in this case the adenine. And if you look at the crystal structures, you will see, and we noticed incidentally, that most of these structures have either this particular residue that I told you about mutated, or it's pointing somewhere else. And at that point, there is the flipping of the base, and therefore the formation of the Uxten base pairing, which is unusual in this type of polymerase. So unusual, in fact, that they were able to publish such paper in very high impact journals. So the hypothesis we generate is that actually this residue here helps the correct base pair for the Watson and Crick base pair to be formed. So we came up with a collective variable to look at the correct base pairing, and we have the wild type where we have the energy between the two minima, the Watson and Crick and the Uxten, and then you see that the Watson and Crick is much more stable because of the presence of this arginine that stabilize the pairing. If you remove that and then you calculate the difference between the Watson and Crick and the Uxten, then well, basically is actually what is important is the energy of the two states, which become isoenergetic. So you can have either Uxten on Watson and Crick, and that's how then we propose that there is a strategically located residue there which promotes the correct base pairing for nucleic acid biosynthesis in polymerases. So again, based on the observation. One last example, it's this particular lysine here in this exonuclease, which is believed to be deprotonated because it needs to act as a general base to get the proton from the water. This is a water that will attack on the phosphate. And what we found is that indeed when this is protonated after the protonation, after the protonation of the water molecule, then you form a cation pi interaction with this conserved tyrosine on the side. At that point, this interaction allows the tyrosine to bring out so there is an oscillatory dynamics here of this couple, and that prefers to go out in such a way that the lysine can release the proton in solution and be back again to reinitiate the cycle. So you see here the simulations where the lysine is in neutral, prefers to stay next to the water, of course, ready to act as a general base. But once we simulate the protonated state, the cation pi interaction brings out the lysine, which then we show through here QMMM simulations, the reaction to happen. And here you move the proton outside that can jump actually on an aspartate or a water nearby. So in this way, again, we were able to form a closed loop sequence of steps that allows the reaction to happen and to form again the new system once the proton is being released to undergo catalysis. So again, another example of how we have used this information to generate mechanistic hypotheses. Okay, then one more point regards the presence of a third ion. I mentioned this at the very beginning. This was a big finding in the field because there is the 2-methyl ion and now there is a third metal ion. As you see here, it ended up in Science. And we put together in this paper here a table of the PDB that are now showing the third ion present in the 2-methyl ion structures. And there is a particular paper that came out on this human exonuclease 1 enzyme where there is a transitory third ion that is found in the reactant but not in the product. So these are in cristallo studies, which means that we have several crystals along the reaction of the enzyme, starting from the pre-reactive to the post-reactive states. And we run our simulation. In this case, this is the enzyme. And this is what the enzyme does. It cuts the last nucleotide of the 5' strand in case of a process related to repair of DNA and so on. So this is what is called exonuclease, because it cuts the extreme of the strand. Okay, so here we made an observation. When the third ion is present, we have this glutamate that is folded toward the third ion that is here, but when it is not present in the crystal, the glutamic acid points outside towards the solvent. So here we run simulations with and without the third ion. Here you see one of the simulations in the product state. In the reactant state, it is stable there. If we remove it, we see sometimes a potassium ion, sometimes a sodium ion going there. So there is clearly an electrostatic trap that attracts ions from the bulk that slowly diffuse there. Here you have a case where in the product state, we have, however, we do see the third ion going there in this position here. So this is the frequency, the probability density of having the glutamate either out or in, depending on the presence or not of the magnesium. And what basically this says that if there is a third ion, prefers to stay in. If there isn't, then flips out, like looking, fishing for an ion nearby to bring close to the other two. And here is the GOR of the third ion in the wild type, which is here, so it is present. And then if we mutate in our simulation the glutamate to an alanine, then you don't have anymore the presence of this third metal ion. So again, there is a functional role. There is a way to call, to select and recruit this ion, and then we started to hypothesize the possible role of this ion. Of course, we cannot say yet the role on the catalytic step, but what we saw in our unbiased MD is that once the third ion is there in the product state, the nucleotide that has been cleaved at that point would like to leave, starts to leave. It doesn't leave completely because we assume at that point maybe there is a barrier, or maybe anyway it is not able in this time scale to leave. But we clearly see in two of the replica that the nucleotide that is being cleaved starts to go away from leave the catalytic site. This is in the absence of the third ion. The nucleotide, even if cleaved, stays there. It doesn't leave. So at that point, again, we use this reaction coordinate to look at the possible pathway to see the energy for the nucleotide that is now leaving the catalytic site to exit the site completely. And here you see the energy that has been estimated for the two processes in presence, in yellow, it is in presence of the third ion or not. And then you see that, of course, with the counter ion, because of course this is negatively charged, the exit is much more favored. And it fits, it matches the overall K-cut, and therefore is compatible with the process we are looking at. So in this particular case, we suggest the third ion for the physical step of leaving group departure is one of the players. So it helps to shuttle out the leaving group. And this is a movie that should convey the overall mechanism I described. And here you have the protein. Here at the center, you see the two ions. And now there will be the third ion coming in with the recruitment of the glutamate. So again, here we are merging different part of the simulations, so the bulk ions, so the third ion goes there, but now you'll see that you will see the glutamate switching. It's put here, you see it went close to the other two. At that point, we have the hydrolysis. We don't see that. We jump in the post-catalytic state, and here we have an acid sampling simulations. And at that point, you'll see that actually the nucleotide leaves together with two ions. And we are not forcing that. That's one of the simulations what we see, and it passed through the arch, that is a particular arch that is formed there. So in this case, we have seen again this trafficking of a third ion that seems crucial for catalysis. Also in this case, actually, you can extend now the relevance of this particular mechanism, looking at other structures where there is either the outer or the inner conformation. This is another exonuclease, and these are the other very similar proteins where again there is always an aspartate in this case, or and a glutamate here that is, and a glutamate also here that is positioned in the same way in order to likely act in the same way as in exo in the nuclease we have I've shown you. And this is the work of Elisa Donati in my group. Okay, one more example if I have the time to do it.
3-4 more minutes.
Okay, so I'll reach the end. So the last example is the group 2 introns. This is the system that Alessandra introduced Monday morning, and it's very important for many reasons. In general expression, it's an ancestor, as I already mentioned, of the spliceosome, and is proposed as a gene editing tool. And we are lucky to have these very nice collaborations with these structural biology groups that generate structures for us. And here it's a complex structure because it is 360 nucleotides that are folded, and they again form these two metal ion center. These are the two magnesium, and here you have K1 and K2, as you already seen in the past. What, however, is here important is that there is this particular cytosine that is critical for catalysis. Every time is replaced with another base that is not an adenine, the enzyme doesn't work anymore. And what we have noticed, this is simple chemistry, that these are the only two bases that can be protonated. So maybe this is a role. And there is indeed a role for that, I'll show you. And then there is a second observation that the crystallographers did. They trapped the enzyme in a structure that is called toggling state. It's actually a structure where this junction is called a junction at the catalytic site, changes a bit this conformation. And now this is complex to study. On the other hand, it's very local, is at the catalytic site, and we have knowledge of the starting point and the ending point of this conformational change. We don't know, however, what's for, why this is important. Of course, it has to happen, but at some point during the transition from the first step to the second step of splicing. Now, this is the step that has been already clarified by Lorenzo and Alessandra in this nice paper here with the attack of the water. And now we have now to locate this transition here that has been reported in the structures. We placed ourselves in a very good situation because we had access to 4 new crystal structures where this particular cytosine has been mutated. And every time it was mutated, the good thing is that it was structurally intact, the enzyme. So the enzyme didn't break because of the mutation. However, the assays demonstrated that the mutations were less effective in generating the product of the first step of splicing. So we know that that particular cytosine is important. Okay, so what triggers now the toggling formation? So here I make the story very short. We run simulations of the pre-hydrolytic state, the post-hydrolytic state. We have crystal structure of these, and then we cleave what we call the cleave state, which is the reactive system where we cut the bond that has to be broken during the reaction, and we see what happened when we do this. And indeed, we have simulations with the cytosine unprotonated and with the cytosine protonated. And what happens is that when this cytosine is protonated, and we believe this may be, for example, the proton that comes from the nucleophilic oxygen that has to be deprotonated, we have QMMM simulations for that in the paper. What we see is that the deprotonation of this cytosine destabilizes the catalytic site. You have solvation of the catalytic site, and at that point, one of the potassium ion is immediately released. At that point, when it is released, this also happens in the case of the cytosine if it is not protonated, but in our simulation always takes much more time. This is always instead immediate, basically. So we propose that the protonation of the cytosine induces the release of K1, which at that point allows only without K1, you can have the toggling conformation. This is from the crystal structure. So this is just to show you the moment where the potassium ion leaves spontaneously. And at that point, we have a structure that can go, the pink one here is the junction that has to toggle. So we have used adaptive with path metadynamics to look at that point at the structural conformation change of the toggling, going from the pre-reactive through the leaving of the K1. So here, so we have the leaving of the K1, and at that point, we have the process that can take place, and this is useful to go to the second step of splicing. So that's the positioning of the toggle conformation that they were able to observe in crystals. And the energy is compatible again with the K-cut, And therefore, this could be a possible explanation and visualization of the transition between the first step and the second step. And this is the work of Jacopo in my group that just came out a few weeks ago. Okay, so I'm finished. I hope I was able to show you how to integrate the view of structure, dynamics, and function for catalysis, in this case on RNA and DNA. We have generated new mechanistic hypotheses based on the role of key residues. We have expanded the functional implication of the 2-methyl ion to a second shell that is now recognized to be important. And then you see now trafficking of cations at the reaction center is also important for this type of reaction that are conserved in many enzymes. With that, our direction now, of course, is to translate this information, I hope, in enzyme design, of course, drug discovery. This is what we are doing now with all this information. And I want to thank the people involved in my lab, all the lab, where you see Pietro, Elisa, and Jacopo, who have been involved in part of the different parts of the work that I show you. And then I thank you and be happy to take questions. Hello? Alessandro?
Alessandro, you have to unmute. Yeah, I'm muted. Sorry. It was. Thanks very much, Mark. It was a very clear talk. There are already a couple of questions on the chat. I start with the first one. For the simulations, you use Amber both as force field and as program, or you perform the simulation with GROMACS, OpenMM, or something else?
Yeah, usually we use Amber as a force field, but then we perform simulation with GROMACS. There are people in my group that also like to use Amber time to time, but most of it is done with GROMACS. Mm-hmm.
Then there is a second question, which is also related to a question which I had myself. This is a question from Ali. Standard GGA functionals in DFT are well known to make hydrogen bonds very sticky due to the lack of dispersion. Can you comment a bit on the role of treating this interaction correctly? And how would using a different functional eventually change the results? Very difficult.
That's a very good point. So I have to say that, of course, this relates to catalysis at the QM level. So we are bound to the fact that in the methods we use, typically Carparinello, as I mentioned, the level of theory is DFT. The functional is most of the time DFT, even though now there is the implementation, but it's very super costly, so not really affordable at the better functionals. But of course, now in our case we are lucky in a way. If I'm not wrong, you said iron, right? The metal it was mentioned. So that are more difficult cases because you have the open spin system, and therefore you have to care about the oxidation states. In our case, we always have closed systems. So magnesium, calcium, and are not do not present this challenge. So in a way, we are lucky in that case. Otherwise, I think yes. If
so sorry, Marco. I wasn't even thinking of those types of exotic ions. I mean, just standard hydrogen bonds.
Yeah,
are known to be very sticky, and the standard trick is to include these semi-empirical corrections like Grimmer dispersion correction. So it's with essentially zero cost, and this helps. So I'm just curious about whether this has been checked or whether this is known, understood, how much it affects things.
Let me ask you back, because I'm just for me to understand whether
Yeah.
Are you also considering the same empirical methods you would suggest, or it's a correction that you would?
No, yeah, it's a post DFT empirical correction.
No, we haven't checked ourselves these effects, to be honest. At the end, this is a well studied, and it's giving the results we care. So in the sense that it matches the barrier, we know the barrier at times is underestimated, and that's sometimes accepted. especially, you know, if you deal with the proton transfer, then you also have other type of questions related to how that happens and the barrier for it. But yeah, the short answer for you is that no, I don't know about these corrections enough to tell you if would help or not in the scheme we have.
All right, thanks.
Yeah.
Well, I have actually a question which is related, which is in a way how much you can trust classical molecular dynamics when you start having ions in the business, in the sense that I remember from the old times when I was also doing similar things, it was always a little bit of an issue deciding how to treat the metal ions. Now things are better. You can trust the force field, at least qualitatively. How does it work?
Yeah, I do see the challenge. I'm well aware of that. I would divide these in our case in two different problems. We have the problem of the catalytic ions, which are in the site and stay there. And that's a problem related to the stability of the coordination. And we showed in the past that if you redistribute the charge of those two ions around the ligands, there are ways in which you can stabilize these two octahedral coordinations, so the two ions. So in a way, if you have a good crystal, then you stick to that coordination and you are fine, because in a way, you assume that there are no ligand exchange apart from the leaving group that is leaving. I would say it's more challenging when you have this binding and unbinding event of ions, because in that case, well, first of all, this diffusion of ions is not easy to see in, at least to this scale we care, in plain MD. And then you also have a ligand exchange, the solvation, and that is, I would say, more challenging. Now, what we do, we use the parameters for those ions, those that are in the solvent. We use the parameters that are now available, the Merz or the Kalman parameters. And of course, what we don't care at that point very much on the coordination of the ion when it comes and goes, because we know that it's going to be very flexible, but we care about the location and how many times we see this happening in our simulations.
All right, so we still have two questions. Yeah. Maybe we can move to the next two questions, because otherwise we'll run short of time. Next question is, how do you derive a most probable mechanism from bias simulation methods like metadynamics?
Well, that's a very interesting question, because at the end, you need to know the reaction and use your chemical intuition to come up with a reaction coordinate that describes the chemical event under investigation. So if I have an SN2 reaction, most likely the bond information, the length of the bond information, or the length of the bond in breaking, or the difference between the bond information and breaking, some combination of the degrees of freedom of the system should describe your reaction. That's how we start with. And then it's a trial and error process. So you start with a reaction coordinate or a collective variable, and you see if that leads the system where you want with a decent energy profile. And then you repeat this till you find the correct reaction coordinate, which is never the true reaction coordinate most of the time. It's a approximation that allows you to capture the chemical event you are interested in. And that's how we start, basically, and then we refine this.
Okay, so we now take the last question, which is, well, I saw in the last example you used the 12-6 Lennard-Jones model for the divalent metal ion. Do you think that the use of 12-6-4 Lennard-Jones model developed by Lee et al. Could produce different results?
So yes, I know these new models that came out from Merz. These are very recent, and now actually they are available in Amber. And at that time we used what we had. Now this is a new model. I can't answer if this would change or not. Of course, we have to try in the future to implement also now these new parameters for the ions and see. I tend to think, maybe I'm optimistic, that yes, we may see some differences in the energy, but not so much to, you know, be, at the end is a qualitative match. What we look at is not really, we want to, in fact, we can't really match with the decimals, the barrier that we see. We don't know exactly if that barrier is for chemical step, physical step. So the answer is no, we haven't tried. And yes, we have to try. And of course, it will be better, most likely. The overall picture, I would say, won't change much, but of course, it's interesting.
All right, so this was the last question, and it's also the last talk of this morning session. So I would like to thank both speakers again for their very clear and interesting presentations. We reconvene this afternoon at 2 p.m. So see you this afternoon. Thanks very much for following.
Bye.
Bye-bye.
Bye-bye.